integrated pest management. So I studied this actually back in the 70s. I worked in integrated pest management. And integrated pest management is a really cool field because how many of you know Silent Spring, Rachel Carson, Cesar Chavez? These were some of the leaders in this field, right? You know, and so this I bring into the kids. I say, you know, back when we were using DDT, you know, the eagles would sit on their eggs and crack them because of the use of DDT. And Rachel Carson brought this, you know, to the forefront. The use of DDT everywhere was killing the insects. It was disrupting the food chain. And so she really started the environmental movement. And from that, I actually was working in California on integrated pest management when Cesar Chavez was doing his work. The state of California was looking at the 10 most used pesticides in fields because farm workers were dying. They were coming back poisoned. And so I did the study on carbaryl, on 7, to look at the mutagenic, the carcinogenic, and the teratogenic effects on it. So Cesar Chavez was another wonderful leader. So these, again, you know, are avenues for your kids to explore, right? So integrated pest management, it's a system that uses the least toxic methods for control. And for school gardens, that's really great because we can't use toxins in the school garden. We don't want to use them in the school garden. So it's based on, you know, the ways that we're managing things. It's based on cultural uses, cultural practices, and it's a pretty fun thing. So when I looked at, okay, we need to develop an IPM, that's kind of the short word for it. We need to develop IPM for control of slugs and snails for school gardens. So I just went to UC Davis. And I looked at UC Davis and started looking at, you know, what they're using. So again, you know, this is just slug and snail damage is a big problem in agriculture. Farmers have been dealing with slugs and snails for a long, long time because they cause a lot of damage to crops. These are papayas. This was giant African snails rasping on papayas. And so they, you know, people might still buy them, but you don't have that beautiful, perfect fruit anymore. So what we did is we went to UC Davis. I went to the UC Davis website and looked at what they were doing, and they recommended the use of shelters. One, clean up the garden, get rid of anything laying around where they might be hiding, because what are things that slugs and snails like? What are the conditions that they like? Moist, dark, right, moist and dark. So you look and you find places where moist and dark where you don't want them, and then you make moist and dark places where you do want them. So a lot of times we'll do a hunt, you know, we'll hunt through the garden. We'll do this in a little bit. And you start to kind of think like a slug or snail is what I tell the kids. Think like a slug or snail. Where would you want to be? You know, they're going to be more active in the evening, so in the daytime they're going to be hiding mostly. You know, if it's rainy out or cool out in the morning, you might see them out and around, but mostly they're going to be hiding. We always really push safety. So we're always saying, you know, when you're out there doing your collection, you're wearing gloves. You know, this was a big thing. You know, we had people saying, well, you're going to teach this to the kids, you know, the safety issues. And it was like, they can go home and get this. So let's teach them how to be safe at school. So we always say, wear gloves. When I'm in the lab, I've got gloves on. If I'm working with the live parasites, I've got a mask on and I've got my glasses on. If something splashes in, you know, it's like, no. So PPE. So we wear gloves in the garden. And then these are some examples. So this was from the pilot study I did. We tested 5 different types of shelter material. And I did this in 5 schools. We had 2 sets of 5 different types of shelter materials that we placed in the garden. We ran this as a regular science study. So we made an insulated tarp. It was a reflective tarp, and then I got a little thin piece of fiberfill. And then we used black plastic for underneath, and we taped it with duct tape. That was one. We had a raised wooden board. We had cardboard. We had a piece of black plastic, and we had weed cloth. And then we would go out in the garden I would come to the schools once a week, and we'd go and look under the shelters and see what we found. At one school, Kanuaka'aina, they had so many snails up there, and they were destroying the mamaki, they were destroying the native plants. That's the only school we did not use this methaldehyde. There are very few baits that can be used at schools, but there is one bait, and it's not Sluggo. You have to actually say that it is approved for school use. So during the Christmas break at that school, I did use bait because they were everywhere. They would crawl up the walls. If you had something posted outside, they were eating the paper off of, you know, postings that were on the door. They were all over the sides of the building. They were smashed on the ground, and you know, whether they were infected, none of them came up infected, but we only tested like less than like 0.1% of them. So we really don't know, but they were damaging the native plants for sure up there. The tea leaves, the mamaki, the kupu-kupu, they were all over the place. So in that case, we did use a bait just because it was really, really heavily infected. But that's the only place we did. And we really, you know, promoted safety. The kids always worked in teams. So when we go out there, you know, we had our clipboard and there was a data collector. Now that data collector maybe did not wear gloves because, you know, handling the pencil. Your kids always are wearing shoes in the garden. Never pick them up with your hands was one of the things. So we used tongs or chopsticks to pick them up. And then we make this slug jug. This is how we get rid of them. We make a very, very saline slug jug, and we'll do that later this afternoon. We use tongs or chopsticks and put them in the slug jug. And then I always tell the kids, you know, you kind of want to watch, and it's like, don't watch when you put it in so you don't get anything splashing back on you. Wash your hands when you're finished. For people that are weed eating or working around a garden, there's people who have hit slugs and snails and had that hit them in the face. So people now on the Big Island, a lot of them, if they're weed eating or something like that, they're wearing a face shield, you know, just to prevent that. There was a woman on Maui who said, I just got hit by slug slime again. The other thing too that we're finding is garden hoses. A basketball player from UHH is pretty sure he got infected by drinking from a garden hose. Slugs will crawl up into them. So we tell people, put a nozzle on the end of your garden hose. Put something on there that they can't get in through. So that's another thing we tell people, you know, don't drink from a garden hose. Okay, so what we will be doing after a little break is we're going to be going out into the garden and we're going to go do a slug hunt. It's summertime. We did have rain a few mornings ago, so we may find some. I'm hoping that we will find some. And what we're going to do at that time is we're going to, anything that we find, we're going to put into a Ziploc baggie so that we can look at them and then start to talk about biology and morphology and identification and that kind of thing. Did you guys bring garden gloves with you perchance? Anybody? No? Okay, so we'll just make sure that we use chopsticks. Now sometimes I'll be out and it's like, man, I don't have my gloves on and there's one. So what I'll do, and actually a lot of times we have traps set up around our lab, and so I go out and collect regularly. You know, you take the Ziploc baggie and turn it inside out and grab it. So, you know, there's a lot of little things that you can do if you're in, you know, you can take a leaf and grab it. The snails, you know, the European garden snails, they're a little harder to grab with chopsticks and that kind of thing. The tongs are easier, but they were up under things and that kind of thing. And I was working with the senior class on that. So we were just being very careful. Everybody was wearing, you know, latex gloves, waterproof gloves. And so, you know, a little bit more dexterity. But with the younger kids, I really, really push like, wear gloves, wear gloves, wear gloves. Don't touch them, don't touch them, don't touch them. But we will go out and we will take a hunt. And so let's just have a little bit of time for discussion right now.
I mean, I know the animal, but what is the European?
The European garden snail is, got lots of pictures, and it's actually in your book, at the end of the book. Okay. It's kind of a more roundish shell. It's banded. It's got some coloring. It's got a brown shell, and it's sort of banded. Let's see which page. Page 109. It looks very similar to another snail called Bradybus similaris. I forget the common name of that. Again, this is where I'm going to really rely on the people that I know that are malacologists. Noreen Young at the Bishop Museum, she's fantastic. And it's like, Noreen, could the bishop make this be part of your job? Because we're going to be sending you and asking you a lot of questions. A lot of the different slugs and snails, you actually have to dissect and look at the reproductive organs to be able to figure out what they are. So identification, and and we'll talk about this afternoon, is going to be like, when we would get a snail that we didn't know, we would just say snail, and then try to describe it and take a picture of it. And so there's going to be some of that. It's going to take a while to be able to identify what's on each island, who they are. Yes?
So I have a mamaki plant, so it looks like something's eating it. So is that a snail?
Probably. It could.
Yeah, I haven't really been paying attention, but I'm like, what's eating,
Yeah.
Okay.
Yeah, well, we relocated to Newport Beach because my son went into an acquired brain injury program, and I always plant food. It doesn't matter if I'm in an apartment or where. And I can't believe how many slugs there are in a little apartment yard less than a mile from the coast in California. I kill them all the time, and they have black rats there. And so I'm actually going to be working with California pretty soon, doing a presentation to say, you need to be looking out for this. So, yeah.
What are the preferred snails for escargot?
The European garden snail. That's the one that they raise for escargot. Yeah, that is the one.
They are potential carriers also.
And you will find them in the garden. So we,
Worm, but they are a carrier.
yes, and so that's why I include them, because we will find them in our gardens. And so we're removing those too. So we do collect that data on them as well. Yeah, and those guys were actually introduced, the black one, the Platydemus maniquari. When the African snail came in, population went nuts. How did the African snail get here? A woman brought 2 of them in her suitcase to Oahu and thought they were pretty and let them go in her garden. A guy on Maui ordered some through the mail to use them for medicine. That's how the African snail got here. And then they just, the population exploded, and farmers were like, ah! And so what happened? Biocontrol gone bad. It was like the mongoose situation again. Bring in the mongoose for the rats. Somebody didn't do their homework. There was so much pressure on the state that we need to control these, that there was evidence in the Pacific Islands that the predators they were bringing in were not good, that they weren't going after the bad snails, they were going after the good snails. We had amazing species of native snails here in Hawaii, amazing species. Many, many, many of them are extinct. Many, many of them are threatened. Up here over in the mountains by Waianae, you have a really, really cool native snail program going on. There's a native snail breeding program going on through DLNR and the university, and they have really elaborate snail enclosures on these few trees that the native snails are living on up there to keep the rats out and to keep the predatory snails out and to keep the Jackson chameleons out, because these guys are all going after our native snails. And so they actually brought in Platydemus maniquari to try to control the African snail population. They brought in the rosy wolf snail to try to control the African snail population. Instead, they went after our native snails. So that's why you got to do your research.
Does
Yes.
the mongoose get affected?
That's one of the animals that Chris Neubauer is looking at, and I'm interested to hear what he has to say, because I hear from our lab we're running the samples and they're coming up on the PCR, but they're not sure if they're not coming up the same way that we usually have stuff come up on our PCR results. So we're not sure yet what is with the mongoose. But for many, many, many conferences that I've been to, the people there have said, no, mongoose can't get it, no, mongoose can't get it. But it's, we've got so much here that it's like, let's make sure that the mongoose can't get it. So that question might be answered pretty soon, I hope. Good question.
Also,
Take one over.
It's easier
That's all
for another slug population to grow because it has less competition.
Actually, what we really see populations fluctuate a whole lot, and malacologists don't even know why. So one year you might have a whole lot of African snails, and then the next year they're not, and you have a whole bunch of leatherback slugs, and then the next year you have a whole bunch of semi-slugs. And so nobody's really sure why those populations are fluctuating. And again, that's a whole another great thing for kids to be collecting data. We can actually start to see what is happening over a long term, and that's really, really important. So is it that disease are affecting them? It could be, you know. I mean, you know, the semi-slug, we vilify all of these creatures, you know, like, but it's like, it's not their fault that they're getting this, you know, it's not their fault that they're getting this, but does the disease affect them? You know, we see very strange behavior with the semi-slug. I've seen them climbing right up the door and underneath the door handle. I've found them inside of my kitchen. You know, it's like, how did they get in? There's a tendency amongst parasites to change the behavior of your host animal. So is that happening with this? I mean, when you have a semi-slug, and literally, these guys can have thousands of parasites in them, you know, 3,000, 4,000, 7,000. I think the highest we've hit was 7,000 so far. Does that change? Does it affect their health? Does it affect their reproduction? Does it affect their behavior? We don't know, but it could, you know. It's a possibility. It's like the, I forget the disease where the mouse will actually go right up to the I've got it right. I think it's the one that the monk seals are getting. You know, the mouse is infected with it, and it will go right up to the cat because that's part of its life cycle. It has to go through the cat host. And so, you know, it's interesting, this mouse will basically offer itself to the cat to be eaten so that that life cycle can continue. Parasites are fascinating. There's a wonderful book called People, Parasites, and Plowshares. And it's absolutely fascinating when you think of this microscopic creature that has this innate intelligence. You know, it really does. It has this innate intelligence to finish its life cycle. And it's really quite remarkable. I mean, you have to really marvel at life. That's all I can say. It's a great book, a great read. Yes?
Just wanted to share.
There is a native snail extinction prevention program in Hawaii. They helicopter people in and do all kinds of cool stuff. I mean, it's cool. Our native snails are beautiful. People came from around the world and collected them. That was, you know, we had collection, habitat loss, and predation. That's what accounted for the loss of our species. You know, and these are really, really unique snails. And they figure probably, I don't know, there's maybe a third of the different species left. That's how much extinction that we've had of our native snail species. So, and native plants too. These invasives are really, really hard on native plants, in particular the endangered plants that they put out. Stephanie Joe had a really good paper that she put out on it and talked about, there was something like, you know, 50% loss of these, you know, endangered plants that they were putting out to replant. So these invasives are, you know, problems. So there's a lot, that's what I like about this curriculum. There's lots of avenues to go through, you know, and that's where I say this is a template. You know, it's not, it is rat lungworm, but you can really allow students to go off into independent investigations because there's so many different areas to go through from this.